So that last one got cut off. I apologize. Going back to where we were at. When you snap off that end phosphate, then it is going to become ADP. And that just indicates that that last phosphate has been broken off so that energy could be released and the body can ultimately put that energy to use then. If you take a look at the slide that is called glycolysis, so glycolysis is really telling by its name. If you look at the front part, it's a prefix. The last part of the word is a suffix. Glyco means sugar. Lysis means to destroy. So anytime you see glyco attached to any word, you know there's sugars involved somehow. And if you look at lysis, that is to destroy, and that holds its meaning again, no matter where you see that. So we are breaking sugar. And the reason that we're breaking sugar is because through that process, we know, again, from what we talked about earlier, that when you break those bonds, energy is given off. And that initial energy that is going to be given off through the breaking of a sugar molecule is ultimately going to be put to use to build ATP molecules and really start the process of cellular respiration. Typically, glucose is going to be the sugar that we use. So that glucose is broken down. We get that initial energy. We start the process of cellular respiration and making our ATP molecules. And by the time we get to the end of glycolysis, we have 2 ATP molecules. So then we move into the second part of cellular respiration, which is the citric acid cycle. And I put the image on there not because you have to memorize anything, but I want you to understand it's a very complex process. We could probably spend a semester alone just on cellular respiration, but not to get bogged down again in the arrows and the abbreviations and everything else that you have going on there, but just to see that you have a lot of changes as these molecules make their way through the process. The reason we refer to it as the citric acid cycle is because it does happen in a cycle format. So the molecules would go through the process once and they would produce 1 ATP. Then it would go through the cycle again and produce another ATP molecule. So by the time you get to the end of the citric acid cycle, you have 2 new ATP. So we got 2 ATP molecules in glycolysis, we got 2 in the citric acid cycle, which isn't too bad. But when you consider the fact that our body is trillions of cells in size, 4 ATP molecules is not really going to be enough for us to sustain our energy needs. So now we have to go to something a little bit more intense, and that's going to be the electron transport chain. So what happens is through glycolysis, through the citric acid cycle, yes, we were building some ATP molecules, but ultimately what we were doing is we were organizing and positioning all of these electrons to go through this process of the ETC. And it's equivalent of taking a slinky and putting it at the top of the staircase. And then you push it over and on down it goes. So we've been organizing these electrons, getting them moved to the beginning of the electron transport chain, which is very much like a staircase. And then in order to get them to come down the staircase, we have to have some sort of strong pull. And that's where the oxygen molecule comes in. So you inhale, bring that into your lungs, and now it's going to use its strength to pull those electrons down the staircase, so to speak. And every time an electron moves down a step, it actually gives off energy. So as it moves through the process, giving off energy every time, we ultimately will harness that energy and build ATP molecules. So by the time we get to the end of the electron transport chain, we have built 32 ATP molecules. It completely blows the other 2 out of the water. So glycolysis, we get 2, Krebs, we get 2, and now in the electron transport chain, we bump that up to 32 molecules by itself. So that oxygen is huge because that has such a strong pull. It can get those electrons down the staircase so they give off their energy, which we ultimately use to build our fuel source. Now, if you start working too hard, if you start running, you'll notice that your breathing rate increases because your body is burning through the ATP and you're trying to replace that ATP. Well, you need to have oxygen in order to do that. So as you burn more ATP, we need to breathe harder to get that oxygen positioned into our cells so that you can keep the process going. In a case where you work harder than what your body can get that oxygen back into your cells, then you will go through a process known as fermentation. So you're pushing yourself really hard, can't get enough oxygen in fast enough. Now we're going to go to burning sugar. Now, when we use oxygen, we're designed to get rid of the waste product when we use oxygen in the electron transport chain, because the waste product is going to be carbon dioxide. You guys exhale, you get rid of the carbon dioxide, and it's fine. We are not designed to get rid of the waste product that we make if we have to use fermentation to supply ATP to our cells. When you burn sugar, it creates something called lactic acid. We're not designed to get rid of that, not easily anyways. And so that lactic acid will actually build up in your tissues. And that's why after a hard workout or you lift for the first time after several months, you have the sore muscles. You are actually feeling that lactic acid that is built up in your tissues, and they will remain sore until your body is able to eventually work it out. The last slide that we have talks about the structure of DNA. And this is a little bit of a beginning for our cell discussion. So when we look at DNA, DNA is ultimately the blueprint for how to make you. This is all the information for how to make or how tall you're going to be, your eye color, your hair color, all of that information is embedded in your DNA structure. Now, DNA is going to be made up of tiny little pieces that are known as nucleotides. And the interesting thing about nucleotides is there are 4 of them. You have thymine, you have adenine, cytosine, and guanine. And usually we just refer to them by their first letter designation. So we use the A, the T, the C, and the G. In living organisms, those are the only 4 nucleotides we have ever found. So every living organism uses those 4 for their DNA, no more, no less. And so when we look at the construction of DNA, what ultimately happens is we get a little bit of a pattern. It was noticed very quickly that when you analyze DNA, your A nucleotide always matched up with your T. Your C nucleotide always matched up with your G. And it didn't necessarily matter the order between the two. A could be first and then T across from it, or T and then A, and vice versa with the C and the G. So as long as the 2 were connected to each other or across from each other in your DNA strand, that was all that really mattered. Through discovery, what they realized is that your nucleotides are actually different widths. And the reason that A and T only match up together and C and G only match up together is because on your DNA, you have 2 backbones, 2 side pieces. And it's kind of like a staircase. When you put A and T together, they were the exact width of the backbones of your DNA strand. And likewise, C and G were the exact width to fit between the backbones of your DNA strand. And that's why we see that pattern of A and T sticking together and C and G sticking together. So DNA made up of those 4 nucleotides, they will basically create a step or a stair that will be linked between the 2 backbones of your DNA. Now, if you look at A and T, and if they are linked together, that is going to be what we call a base pair. C and G would be another base pair. And these base pairs will link one on top of the other down the strand of DNA. The human DNA strand is going to have 3 billion base pairs. So from the top of the DNA strand to the bottom of the DNA strand, you are going to have 3 billion of these base pairs that are stacked with one another. That's incredibly long. We know 3 billion is a big number, but in actuality, you can't really even wrap your head around what 3 billion is. So we have this incredibly long DNA strand. And if you want to consider how amazing the human body is, we took something that was 3 billion base pairs long and found a way to wrap it and weave it so that it could pack itself not just into your cells, but into a smaller component of your cell, which is the nucleus. So your DNA strands, when we look at them, they have a spiral helix or a spiral formation to them. And that's the reason why, is because we would not be able to pack 3 billion base pairs in if it was just a straight long chain. But by wrapping it and having it take on a spiral shape, now we can get it to fit in a way that we can compact that and put it into the nucleus of all of your cells. So that takes care of the lecture side of things that we need to talk about. Once you have your notes and everything completed from the video, you can move on to the Testmos. Strongly suggest taking the Testmos after you've watched the video and picked up that information. And you only have one attempt at the Testmos. So please make sure that you wait until you are ready. You have until 11:59 on Sunday evening to get the Testmos done. If you want to go back and look at stuff in the video, that is perfectly fine. You can use your notes on the Testmos. That works as well. The only thing that I am asking on the Testmos is that you guys do not get together in groups and take it collectively. So you get one submission. Please take it on your own, but you have access to your textbook. You can look at pieces of video again. However you want to do that, that is perfectly fine. So watch the video, take notes, and then go ahead and take that Testmos when you feel like you're ready.